Okay, so I wrote an outline for chapter 20, quite a bit of it. Cindy Owen really covered well in the discussion board, so I'm going to just briefly go over the basics, and I'm going to go more in depth for the things that weren't talked about in the discussion board and reference what can be found on the discussion board. And then I will be posting a link to my own outline. The anatomy of the heart. The location of the heart. The heart is about the same size as your closed fist, about 250 grams mass in adult females and 300 grams in adult males. It is located on the diaphragm near the midline of the thoracic cavity in the mediastinum region. About two-thirds of the heart lies to the left of the midline, with the apex, which is the tip of the left ventricle, the lower chamber of the heart, directed anterior, inferior, and to the left of the diaphragm. The base of the heart is its posterior surface, formed by the atria, or the upper chambers, mostly by the left atrium. Several other distinct features of the heart include the anterior surface deep to the sternum and ribs, the interior surface between the apex and right border lying mostly on the diaphragm, the right border facing the right lung extending from the inferior surface to the base, the left border, pulmonary border, which faces the left lung and extends from the base to the apex. The pericardium is the membrane that surrounds and protects the heart. It's divided up into the fibrous pericardium and the serous pericardium. It confines the heart to its position in the mediastinum while allowing freedom of movement and rapid contraction. The fibrous pericardium is superficial, composed of tough, inelastic, dense, irregular connective tissue. It attaches to the diaphragm with its open end fused to the connective tissues of the blood vessels entering and leaving the heart. It prevents overstretching of the heart, provides protection, anchors the heart to the mediastinum. Near the apex of the heart, it is partially fused to the central tendon of the diaphragm, thus movement of the diaphragm facilitates movement of blood by the heart. The serous pericardium is thinner and a more delicate membrane, forms double layer around the heart. Outer parietal layer is fused to the fibrous pericardium. Intervisceral layer, or the epicardium, is one of the layers of the heart wall. Between the parietal and visceral layers is a thin film of serous fluid. This secretion of the pericardial cells, or pericardial fluid, reduces friction between the layers of the serous pericardium as the heart moves. The space that contains the few milliliters of pericardial fluid is called the pericardial cavity. There are three layers of the heart wall: epicardium, myocardium, and endocardium. You can read about these in either my outline, the outline provided by Dr. Bennett, or the discussion board. As well as you can also find the chambers of the heart. Great post by Cindy Owen with the right atrium, left atrium, right ventricle, and left ventricle. That would be the four chambers. In addition to what Cindy posted, the auricle on the anterior surface of each atrium increases capacity of the atrium slightly. Sulci on the surface of the heart contain coronary blood vessels and variable amounts of fat. Each sulcus marks external boundary between the two chambers of the heart, or between two chambers of the heart. Deep coronary sulcus encircles most of the heart and marks external boundaries between the superior atria and inferior ventricles. Anterior interventricular sulcus is a shallow groove on the anterior surface of the heart that marks the external boundary between the right and left ventricles. The posterior interventricular sulcus marks external boundary between ventricles on the posterior aspect of the heart. Interatrial septum between right and left atrium. And note the fossa ovalis also involved in that, which you can find in the book. Tricuspid valve, also known as the right atrioventricular valve. And interventricular septum separates right and left ventricle internally. As for myocardial thickness and function, thickness varies according to function of the chamber. The atria is thin-walled and delivers blood under less pressure to the ventricles. Ventricles are thicker due to pumping blood at a higher pressure over greater distances. The right ventricle pumps blood to the lungs, thus has a smaller load than the left ventricle. which pumps blood to the whole body. And so, furthermore, the left ventricle has a thicker wall. The lumen of the left ventricle is circular white, while the lumen of the right ventricle is crescent-shaped. The fibrous skeleton of the heart. It is formed by dense connective tissue, consists of 4 dense connective tissue rings that surround the heart valves, fuse with one another, and merge with the interventricular septum. It forms structural foundation for the heart valves and prevents overstretching. And it also serves as insertion point for bundles of cardiac muscle fibers and acts as electrical insulator between the atria and ventricles. For heart valves and circulation of the blood, I posted a video previously, or you can find more information on page 767. Systemic and pulmonary circulations. Cindy Owen put a great post in the discussion board. There wasn't much to add to it. As for coronary circulation, which was not included, but was in the book, is a network of blood vessels for the myocardium, also known as cardiac circulation. Coronary arteries branch from the ascending aorta and encircle the heart. These arteries are squeezed shut during contraction of the heart. When the heart is relaxed, high pressure of blood propels from the aorta through the coronary arteries into the capillaries and then into the coronary veins. Coronary arteries. There are 2 coronary arteries, right and left, which branch from the ascending aorta and supply oxygenated blood to the myocardium. The left coronary artery divides into the anterior interventricular and circumferential branches. The anterior interventricular branch, or the left anterior descending artery, or LAD, LAD, is in the anterior interventricular sulcus and supplies oxygenated blood to the walls of both ventricles. The circumflex branch is in the coronary sulcus and distributes oxygenated blood to the walls of the left ventricle and left atrium. The right coronary artery supplies small branches, or atrial branches, to the right atrium and divides into posterior interventricular and marginal branches. The posterior ventricular branch follows the posterior interventricular sulcus and supplies the walls of the 2 ventricles with oxygenated blood. The marginal branch is along the right margin of the heart and transport oxygenated blood to the myocardium of the right ventricle. Most parts of the body receive blood from branches of more than one artery, and where 2 or more arteries supply the same region, they usually connect. Connections are called anastomosis and provide alternate routes called collateral circulation. The myocardium contains many anastomoses and connect branches or extend them, the anastomosis connect branches or extend them, and they provide detours for arterial blood if a main route becomes obstructed. Coronary veins. Most of the deoxygenated blood from the myocardium drains into a large vascular sinus in the coronary sulcus on the posterior surface of the heart, known as the coronary sinus. The deoxygenated blood in the coronary sinus empties into the right atrium. The principal tributaries carrying blood into the coronary sinus are the great cardiac vein, middle cardiac vein, small cardiac vein, and anterior cardiac veins. For more details on these veins, you can go to page 770 in your book. Reperfusion. When blockage of coronary artery, it deprives the heart muscle of oxygen. When blood flow is reestablished, it can also cause more damage due to free radicals from the reintroduced oxygen. Enzymes that convert free radicals to less reactive substances include superoxide dismutase and catalase. Nutrients that remove free radicals from circulation include vitamin E, vitamin C, beta-carotene, zinc, and selenium. Cardiac muscle tissue and the cardiac conduction system. There's a great histology of the cardiac muscle tissue from Cindy Owen on the discussion board. And it compares cardiac muscle tissue to skeletal muscle tissue, and including what we learned in A&P Part 1 with the actin and myosin, the bands, zones, and Z-discs. So we're just going to skip over that. And autorhythmic fibers, the conduction system. I added a little more to what Cindy Owen had posted in the discussion board, so it'd be great to view what she posted. But as for what I have to add, the autorhythmic fibers are specialized cardiac muscle fibers that act as a source of electrical activity. Autorhythmic fibers are also self-excitable. They repeatedly generate action potentials that trigger heart contractions. They continue to stimulate the heart to beat even after it is removed from the body. During embryonic development, only about 1% of the cardiac muscle fibers become autorhythmic fibers. Important functions of autorhythmic fibers include the fact that it acts as a pacemaker and forms cardiac conduction system, which is a network of specialized cardiac muscle fibers that provide a path for each cycle of cardiac excitation to progress through the heart. This conduction system ensures that cardiac chambers become stimulated to contract in a coordinated manner. Action potential and contraction of contractile fibers. I have made a post on this, a video, as well as I actually posted what I have typed up, so you can find that in the discussion board. Production of ATP in cardiac muscle. The cardiac muscle produces little ATP needed by anaerobic cellular respiration and relies exclusively on aerobic respiration in its mitochondria. The needed oxygen diffuses from blood and the coronary circulation and is released from myoglobin inside cardiac muscle fibers. In a person at rest, 60% of the ATP comes from oxidation of fatty acids and 35% from glucose oxidation, with smaller contributions from lactic acids, amino acids, and ketone bodies. During exercise, the heart uses more lactic acid. Electrocardiogram, also known as ECG or EKG, is a composite record of action potentials produced by all the heart muscle fibers during each heartbeat. Electrocardiograph, the instrument used to record changes. In clinical practice, electrodes are placed on the arms, legs, and chest, or limb leads and chest leads. Each electrode has a slightly different recording due to its position relative to the heart. By comparing the records recorded, it is possible to determine if the conducting pathway is abnormal, if the heart is enlarged, if certain regions of the heart are damaged, and the cause of chest pain. 3 waves appear with each heartbeat. You can find these waves in the discussion board, thanks to Cindy Owen and her awesome posts. As for correlation of ECG waves with atrial and ventricular systole, systole is the phase of contraction, diastole is the phase of relaxation, and ECG waves predict timing of atrial and ventricular systole and diastole. The cardiac cycle, also a bit of the correlation of ECG waves with atrial and ventricular systole being included, can be found in the discussion board thanks to Cindy Owen. So I won't go over that as well because you can read it. Heart sounds. Auscultation is the act of listening to sounds within the body, usually done with a stethoscope. Sound of a heartbeat comes primarily from blood turbulence caused by closing of the heart valves. Smoothly flowing blood is silent. During each cardiac cycle, there are 4 heart sounds. In a normal heart, only the first and second are strong enough to be heard with a stethoscope. S1 and S2 is what we will call those sounds. The first sound, S1, or lub, is louder and longer than the second sound and is caused by blood turbulence associated with the closure of the AV valves soon after ventricular systole begins. The second sound, or S2, or dub, is caused by blood turbulence associated with the closure of the SL valves at the beginning of ventricular diastole. Since the sound is carried by blood flowing away from the valves, these heart sounds are best heard at the surface of the chest in locations slightly different from the locations of the valves. S3, which is not normally loud enough to be heard, is due to blood turbulence during rapid ventricular filling. And S4, which is also not normally loud enough to be heard, is due to blood turbulence during atrial systole. Cardiac output. There is quite a bit on cardiac output in the discussion board, thanks to Cindy Owen, and she covers pretty much everything. Factors that affect cardiac output include end-diastolic volume, positive inotropic agents, negative inotropic agents after load, and all of that can be found in her post. Regulation of the heart rate. Some of the most important factors that contribute to regulation are the ANS and hormones released by the adrenal medulla. That would include epinephrine and norepinephrine. The nervous system, regulation of the heart, originates in the cardiovascular center of the medulla oblongata. This region of the brainstem receives input from a variety of sensory receptors and higher brain centers, including the limbic system and cerebral cortex. Then the cardiovascular center directs appropriate output by decreasing or increasing the frequency of nerve impulses in both the sympathetic and parasympathetic branches of the ANS. Proprioceptors monitor the position of limbs and muscles and send nerve impulses to the cardiovascular center as physical activity begins. Chemoreceptors monitor chemical changes in the blood. Baroreceptors monitor stretching of major veins and arteries caused by blood pressure. Cardiac accelerator nerves extend out of the SA node and AV node and most portions of the myocardium. These nerves send impulses that trigger the release of norepinephrine, which binds to the beta-1 receptors on cardiac muscle fibers. In AV and SA node fibers, this interaction speeds the rate of spontaneous depolarization of pacemakers and can fire impulses more rapidly, and the heart rate increases. In contractile fibers, this increases inflow of calcium ions, thus increasing contractility. A good side note that I thought was kind of interesting, subtracting your age from 220 provides a good estimate of your maximal heart rate in beats per minute. Just thought that was a cool fun fact from the book. The parasympathetic impulses reach the heart rate via vagus nerves, which release acetylcholine, thus decreasing the heart rate by slowing spontaneous depolarization of autorhythmic fibers. Chemical regulation of the heart rate. Hypoxia is lowered oxygen, acidosis, low pH, alkalosis is a high pH. Several hormones and cations have major effects on the heart. There are more details about these can be found on 784. I'm just going to briefly mention them. Hormones include epinephrine, norepinephrine, thyroid hormones. Side note about the thyroid hormones, a sign of hyperthyroidism is tachycardia. Cations include potassium, sodium, and calcium. Other factors in heart rate regulation include age, gender, physical fitness, and body temp. Exercise and the heart. Physiological cardiomegaly is exercise that leads to enlargement of the heart. Pathological cardiomegaly is heart disease. Help for failing hearts. There's a good table about cardiac assist devices and procedures on page 788 and 789, including intra-aortic balloon pump, or IABP, ventricular assist device, or VAD, cardiomyoplasty, skeletal muscle assist device. Also, cardiac heart transplant replacement of the heart with a normal heart. Development of the heart. I posted a really neat animation video I found on YouTube about development of the heart because I thought it was pretty interesting. Pretty much, the cardiovascular system is one of the first to form in developing embryo, and the heart is the first functioning organ. Disruption of development can lead to congenital disorders of the heart, which means they're present at birth. The heart begins development from mesoderm on day 18 or 19 after fertilization. In the head of the embryo, the heart develops from a group of mesodermal cells called cardiogenic area. Signals from ectoderm cause mesoderm and cardiogenic area to form a pair of elongated strands called cardiogenic cords, which develop a hollow center and become endocardial tubes, which later fuse together into a single tube called the primitive heart tube on day 21. On the 22nd day, primitive heart tube develops into 5 distinct regions and begins to pump blood. From the tail to the head end, which is the direction of blood flow, regions include the sinus venosus, which develops in the posterior wall of the right atrium, coronary sinus, and sinoatrial node. The primitive atrium, which develops into the anterior wall of the right atrium, the right auricle, and anterior wall of the left atrium and left auricle. Primitive ventricle, which develops in the left ventricle, bulbus cordis, which develops into the right ventricle, and truncus arteriosus, which develops into the ascending aorta and pulmonary trunk. On day 23, the primitive heart tube elongates and begins folding. Day 28, primitive atria and ventricles are reoriented to their permanent place. Remainder of the development consists of remodeling chambers and the formation of septa valves to form four-chambered heart. Day 28, endocardial cushions appear, which fuse and divide in a single atrioventricular canal into smaller right and left atrioventricular canals. Interatrial septum and endocardial cushions fuse, and the foramen ovale develops. Interatrial septum divides right and left atrium. The foramen ovale closes after birth. The interventricular septum divides right and left ventricles. The atrioventricular canal, atrial region, ventricular region partitioning completes by end of week 5. Atrioventricular valves form between the 5th and 8th weeks, and the semilunar valves form between the 5th and 9th weeks. Like I said, there's a lot of helpful information in Cindy Owen's post, and I will be posting a link to my outline, which is the complete outline for Chapter 20.